Hi, and welcome to the VieSID Press-release. In this video, we report on articles selected from the dental literature published in October 2023 that we hope might be of interest to the VieSID community. So let's begin. In the Annals of Anatomy, Oshima et al. Described the distribution patterns of infraorbital nerve branches that innervate also the cheeks and upper lips and discuss the risk for injury in case oravesibular incisions are required during surgery. The incision on the upper lip mucosa might result in possible sensory disturbances. Thus, they dissected 9 cadavers to study the gross anatomy of the infraorbital nerves. The study reported the anatomy of the nerve in great detail with an accurate description of the individual variants, which included anastomosis at different positions and communications between the infraorbital nerve and the facial nerve. Here, I will convey only the salient information. Most importantly, Oshima et al. Demonstrated that the distribution of infraorbital nerve branches in the upper lip follows a vertical pattern rather than a horizontal pattern from the lateral to the medial sides. Thus, they concluded that lateral incisions of the upper lip mucosas should reduce the risk of thick nerve injury and thus are preferable to transversal incisions. The lateral incision shouldn't be too deep to avoid damaging the labial glands. The Journal of Dental Research published an article by Yuan et al. Reporting an experimental animal study demonstrating the detrimental effects of a soft diet on the health of the junctional epithelium and periodontal tissues. It is known that the junctional epithelium adheres to the teeth and acts as a barrier protecting the periodontal tissues from microbial ingression. It is also known that the junctional epithelium contains Wnt-responsive stem cell niche that regenerates the epithelial cells. Stem cell maintenance depends not only on chemical signals but also on physical signals. Mechanical stimuli on the periodontal ligament influence the development of its stem cell niche. The authors aim to test whether a similar effect could be observed for the junctional epithelium. A relation between the physical properties of diet and the health of the periodontal tissues has been reported since 1956 and even earlier based on animal studies. The presence of hemidesmosome, which are mechanotransductors at the attachment of the junctional epithelium to the tooth, seems to corroborate this assumption. First, Yuan et al. Wanted to determine when the Wnt-responsive niche establishes in the junctional epithelium, before or after tooth eruption. They use the fluorescent labeling technique to stain the Wnt-responsive cells. In this way, first they demonstrated the presence of stem cells in adult mice up to 300 days after labeling. Then they showed that stem cells niches are not present in pups before the eruption of teeth. Thus, they concluded that the stem cell niche establishes after the eruption of the teeth. Then they conducted experiments to test whether mastication has a tropic effect on the junctional epithelium and its stem cell niche. To this end, they divided the mice into 2 groups, one fed on a hard diet and one fed on a soft diet, approximately 10 times less mechanically challenging than in the control group. This dietary regime was maintained for either 7 or 14 days. At 7 and 14 days chase, the mice on a hard diet showed a significantly higher number of stem cells than the mice on a soft food diet. In a second experiment, the mitotic activity of the cells was detected, revealing that the mice on a soft diet showed a reduced number of actively dividing cells. Thus, the authors proved that stem cells reach full potential thanks to the mechanical stimulation following mastication of hard food. Yan et al. Further tested the effect of soft diet on the health of the periodontal tissues. The soft diet group showed a decreased amount of E-cadherin, a protein for cell-to-cell adhesion. They showed that compromised junctional epithelium barrier functions were associated with mild inflammation, with increased presence of macrophages, phalloid, and interleukin. An effect on alveolar bone crest was shown with a reduction of its height and bone density in the soft diet group. Following a soft diet, signs of periodontal infection and bone deterioration were observed. This article is relevant in shedding light on the importance of proper biomechanical strains on the masticatory system, thus confirming literature reports of the last century. In the Journal of Dental Research, we found an interesting article by Wang et al. On the pathogenesis of COVID-19 related taste disorder and its possible treatments. Approximately 765 million people have contracted COVID-19 worldwide. About 45% reported taste impairment among the symptoms, and 5% of them reported persistent taste loss or abnormalities, also called dysgeusia. This means that approximately 17 million people have had permanent taste perception problems. Sense of taste is detected by taste buds and converted into gustatory signals that travel to the primary taste cortex. They are present in the tongue, palate, and epiglottis, for example, and consist of clusters of columnar taste receptor cells, or TRC, possessing microvilli with receptors for detection of taste molecules. There are 3 different types of taste receptor cells conveying the different tastes: sweet, sour, bitter, salty, and umami. Taste receptor cells are constantly regenerated by the differentiation of stem cells at taste buds. The detailed mechanism of COVID-19 related dysgeusia remains unknown. This could be attributed to the peculiarity of flavor, which is a central combination of senses such as smell, taste, somatic sensation, and psychological state, and cannot be just considered as a disturbance of the olfactory sense. Wand and colleagues describe various hypotheses on the pathogenesis of COVID-19 related gustatory dysfunction, which are summarized in the image reported here. The first hypothesis is that SARS-CoV-2 invades the taste receptor cells and replicates, directly damaging the cells. Moreover, cytokine activity causes secondary inflammation. This mechanism has been proven both histologically and at a biomolecular level, as the taste bud presents multiple types of SARS-CoV-2 receptors, meaning that the virus has the opportunity to bind and infiltrate taste buds. The renin-angiotensin system, or RAS, is a hormone system that regulates body fluid and sodium homeostasis, thus also sodium appetite and preference. The receptor ACE2 regulates also the RAS. Thus, if this receptor is blocked by the virus, the local RAS is damaged and inflammatory factors accumulate, thus disrupting the taste bud microenvironment. Cytokine storms play a key role in the pathogenesis of SARS-CoV-2. They are characterized by high expression of interleukin and tumor necrosis factor and are closely related to the severity of the disease. In taste receptor buds, SARS-CoV-2 S protein interacts with the innate immune receptors, leading to the production of interferon and interleukin and other pro-inflammatory genes. The local inflammation promotes the production of cytokines, upregulating the receptor factors and eventually leading to the cytokine storm. This inflammatory status causes taste receptor cell apoptosis and disrupts the renewal cycle of taste bud stem cells, resulting in prolonged dysgeusia. COVID-19 can cause various nervous system disorders, and in fact, ACE2 receptors are present in the nervous system too. So far, there is no direct evidence of SARS-CoV-2 invading the olfactory center and the peripheral nerves in humans, but there is in mice, although the effects of such invasion were described. SARS-CoV-2 can infect the olfactory neurons in vitro and compromise their function. It is known that dysgeusia results from impaired smell. However, the neurotropic effects of SARS-CoV-2 on taste function is mostly inferred rather than proved. Thus, further studies are needed to test this hypothesis. Zinc plays a key role in cell function and morphology. Its deficiency is associated with impairment of taste buds. The exact mechanism that links zinc deficiency to dysgeusia is not known, but zinc supplementation is proven efficient in clinical settings. Recent development in genomic research have revealed genetic variation linked to the severity of the disease and taste and smell function, offering an interesting perspective on the role of genetic variation in the expression of the disease. Further possible pathogenetic mechanisms were described. SARS-CoV-2 can occupy the binding sites of celiac acid on taste buds, thus accelerating the degradation of taste granules, playing a role in dysgeusia. The ACE2 receptors exhibit significant co-expression with enzymes for the synthesis of dopamine and 5-hydroxytryptamine, which is necessary for the physiological functions of taste buds. Based on the proven or hypothesized pathogenetic mechanism for dysgeusia, several treatment modalities have been proposed, including anti-inflammatory treatment to reduce the inflammatory state generated in response to the virus and thus attenuating the process leading to disruption and apoptosis of the taste cell receptors. Antiviral treatment to inhibit enzyme activity of SARS-CoV-2 main proteases. The use of celiac acid analogs inhibits the combination of virus and celiac acid receptors, at least in animal studies. Zinc supplementation seems beneficial for the recovery from COVID-19 dysgeusia, although it is not yet clear why. Neural protection might be offered by melatonin in case the nervous system is interested, although its effects on taste function must still be evaluated. Finally, taste bud regeneration is promising in case the ability of stem cells to generate new taste buds is compromised. Photobiomodulation therapy with vacuum laser has shown positive results in case reports. This article well reflected the complexity of the pathogenies of SARS-CoV-2 and its possible effects on taste function, which depend also on the individual phenotype of the host. It showed also the incredible medical and technological advancement prompted by the coronavirus pandemic. I would like to mention briefly the article by Verzelar et al. In the Journal of Oral Rehabilitation. The authors had the main goal to develop a set of diagnostic criteria for the assessment of tooth wear. A two-step approach was used to achieve this objective. First, they developed a preliminary beta version of these diagnostic criteria based on the author's clinical experience and supported by a narrative review of the literature. The results of this first step are reported in this paper. Second, they planned future work to finalize the diagnostic criteria with the input from a larger group of experts using an international Delphi process. The authors eventually defined the first version of the diagnostic criteria for tooth wear assessment, which included an anamnesis of the patient complaints, several tools for Axis 1 and Axis 2 assessment of tooth wear-related etiology and effects, and clinical guidelines for decision-making. As usual, the Journal of Prosthetic Dentistry published their annual review of selected scientific literature. that was performed by Kanya and colleagues on studies reporting advances in several fields of dentistry, including prosthodontics, periodontics, alveolar bone and peri-implant tissues, dental materials and therapeutics, occlusion and temporomandibular disorders, sleep disorders, breathing, oral medicine and oral and maxillofacial surgery, caries and cariology. Nature published an outstanding paleogenetic paper by Fo, Ding et al. With relevant clinical implications. Theirs is a fascinating story involving the NAV 1.7 voltage-gated sodium channel, Neanderthals and Latin Americans. NAV 1.7 is a sodium channel crucial for nerve impulse generation and conduction in peripheral pain pathways. NAV 1.7 is highly expressed in nociceptive neurons and regulates their excitability. The gene encoding for NAV 1.7, called the sodium voltage-gated channel alpha subunit 9, SCN9A, expresses mutations that are linked to variation in the perception of pain. For example, family studies have shown that rare mutation in this gene can cause Mendelian forms of either insensitivity to pain or extreme pain syndromes. Zeberg et al. In 2020 showed that Neanderthals carried mutations of the SCN9A gene that generated 3 variants of the NAV 1.7 sodium channels. When all of these 3 variants were present, NAV 1.7 showed reduced inactivation, suggesting that peripheral nerves were more sensitive to peripheral stimuli in Neanderthals than in modern humans. Although it is not possible to infer that they also experience higher levels of pain, as translation of pain is modulated at the level of the spinal cord and brain too. The map showed the allele frequencies of the variant with all 3 amino acid substitutions in modern humans for which the genome was sequenced. These variants are frequent in East Asia and Central and South America. However, individuals carrying these 3 variants are rare in Europe. This high prevalence is not surprising considering the past distribution in Europe and Asia of Neanderthals and Denisovans, another human group closely related to Neanderthals. Through a phenomenon called introgression, which is the transfer of genetic information from one species to another, resulting in hybridization, the Neanderthals variants of the genes were transferred to modern humans. Approximately 15 to 20,000 years ago, modern human populations migrated into the American continent, passing through the Bering Strait. Now, back to the study of Fo, Ding et al. They used modern human samples, the QST cohort of Colombians and the Candela cohort of South Americans. Mean ancestry estimates for the Colombia cohort was 31% Native American, 59% European, and approximately 10% African, which was similar to the Candela cohort. But of course, individual variation was considerable, as shown in this graph, where ancestry distribution is provided by country. The authors tested the allelic frequencies for the Neanderthals variants and found out that this correlated with the percentage of Native American ancestry, as is visible in this histogram. Still, some mutations were carried by chromosomes of European or African ancestry, but the authors attributed this to possible methodological inaccuracy. So this showed that Neanderthal introgression in the genome of Latin Americans. The authors then performed the pain test on the 1,623 Colombians of the QST cohort. With this experiment, they aimed to overcome the limitations of previous work by examining populations with relatively high frequency of the Neanderthal NAV1.7 variants and defining sensory phenotypes experimentally. In particular, they measured pain thresholds to pressure, heat, mechanical stimulus, mechanical and heat stimulus after sensitization with mustard oil, which is an irritant. And finally, they also tested the wind-up ratio, which is a measure of central sensitization pain. They found out that the individuals carrying the Neanderthal mutations showed higher sensitivity to pain, in particular to mechanical pain, where all three Neanderthal variants impacted on pain sensitivity. This illustration shows that with the increase in the number of Neanderthal alleles, the mechanical pain threshold decreases. In conclusion, NAV1.7 Neanderthal alleles trace their origin to the Native Americans that have contributed to the admixture in the Latin American populations examined. As originally shown by Zeberg et al., Neanderthal ancestry in SCN9A gene affects nociception by increasing sensitivity to mechanical and most likely to allogenous pain. This article beautifully shows the importance of considering our evolutionary past in medicine and made it again very obvious how important it is to take into account individual and also geographical variation in clinical settings. We have come to the end of this presentation. I invite you to contact us in case you want to know more about our activities. Otherwise, be ready for next press release. Best wishes to all.